working for the past 1.5 years, looking at Alzheimer's and Parkinson's disease from through the lens of single-cell transcriptomic data. And today we'll try to understand a little bit of the genetic mechanisms that are behind these diseases. So Alzheimer's and Parkinson's are two of the world's most prevalent neurodegenerative disorders, affecting over 80 million people worldwide. And the normal way that people think that these diseases occur is through the accumulation of misfolded proteins, leading to neuronal death. And then in the neuroscience community, the belief is that these diseases have very different etiologies and different mechanisms of progression in the brain. And so Alzheimer's is thought to be the dementia disorder. And Parkinson's, when we talk about it, we generally tend to think of it as a motor disease, which affects different kinds of motor activities. Even the treatment and diagnosis of these disorders are very separate. Research is separate and siloed so far. However, the clinical diagnosis of Alzheimer's and Parkinson's actually has a lot of challenges sometimes, because there is often some overlap that is observed. And even in postmortem analysis of AD or PD patient brains, there's often a lot of overlap in neuropathology. So for example, 50% of Alzheimer's patients, the brains will contain some alpha-synuclein deposits, which is a primary protein aggregate that is seen for Parkinson's disease. And then some of the patients will also have some extrapyramidal symptoms, which are bradykinesia, rigidity, tremor, etc. And then on the other hand, over like around 30% of the Parkinson's patients have some form of dementia, and then more than half, when there is postmortem analysis, they show some kind of tau aggregates, which is again one of the protein deposits which is common for Alzheimer's. So obviously there is some kind of overlap that's going on, and the community so far has been very there's a lot of debate on how this occurs, why this occurs, and so some attempts at looking at the genetic mechanisms was done in the beginning in 2018 by the Brain Consortium. And so this is essentially a correlation of GWAS genes for different kinds of neurodegenerative diseases or just brain-related disorders. And for Alzheimer's and Parkinson's, there is very little overlap, like almost no overlap. And then this study was similar study was done more recently in 2024, published in Nature, and even there the overlap is very less. So essentially, in a neuron review, there was a summary where they kind of say that these are very different disorders with different onsets and progression mechanisms, and there is very little overlap. However, we already have seen some cases where there is some kind of some things that are similar, right? So for the challenge, so we took on this challenge, and then we want to answer what are the genetic mechanisms that are probably causing these observed neuropathological symptoms. So we look at some of the common analytical themes that are present across the studies that people did so far. So the first thing is that most of these studies, like genome-wide association studies or differential gene expression analysis, they look at genes and on a single gene analysis level. So what they're doing is they're looking at each individual gene, and they're seeing if they have any significant association with the disease. And once they find the genes contributing to Alzheimer's, and then separately for Parkinson's, they go and look at try to see if there is any overlap in these genes. However, the problem with this analysis, this technique, is that we know that genes rarely act in isolation. Rather, they have a tendency to work in concert with each other, and they are affected, or they either affect other genes or are affected by other genes. And so this kind of single gene analysis is maybe not the ideal way to look at, to uncover these complex diseases. And the second limitation is that GWAS and DG, both of these methods, don't have that level of granularity that is required, because the brain is especially a very heterogeneous organ. It has around 461 broadly categorized cell types. So GWAS is mostly focuses on the entire genome, and the differential gene expression looks at more or less like bulk RNA sequencing, which has tissue level granularity and fails to account for different cell types and the different roles that they might be playing. So one way around this is to look at single cell transcriptomic data sets. And here the idea is that they have they look at individual genes and they try to record the gene expressions from for each cell type. And then one of the clear indication that this study works is was published in 2019 by Mathis et al. In one of the largest of its kinds, looking at Alzheimer's patient's brain versus healthy patient brain. And they found that APOE, which is one of the major disease-associated gene for Alzheimer's, is highly expressed in microglia and astrocytes. And it's not that much in neurons, even though neurons are the ones which, you know, were thought to be related to Alzheimer's or causing the disease progression. But now the focus is going more towards these other cell types just because we have that kind of resolution. However, we are saying that even the cell type level granularity is maybe not enough to uncover all the technicalities and the different intricate overlaps and genetic mechanisms. So that's because, oh, the font is too small, sorry about that. So this, in the neuron review itself, they did a kind of comparison of disease marker genes across different cell types, and even there they failed to find any overlap between Alzheimer's and Parkinson's in particular. So in our study, we'll focus on these two crucial concepts that we have kind of found so far. So first of all, we'll focus on the fact that genes act in coordination, and any kind of analysis we do will not try to look at genes in isolation, but rather as a whole. And then the second point is that different cells might have different contributions to a disease, and they have different multimodal contribution to a disease progression. Okay, so the transcriptomic data is generally very high dimensional, and as has been already covered by a few speakers yesterday, as well as today by Sebastian, we need to somehow, you know, compress this information into more like tractable levels. And then the common way to do that would be to use like UMAP, TSNE, PCA, etc. However, the problem with this is that doing these kind of latent factor approaches loses the disease context oftentimes, and then the embedding space that is uncovered doesn't have that information that would be there if the disease context was also taken into account. So for that, there could be, so yeah, it's blind to disease context. And then, so to go around that, there could be some supervised predictive algorithms that could be used, but the problem there is that they lack the interpretability that comes from learning a compressed representation of several sources of gene activity variation. And so to bridge this gap, what we are doing is we are using partial least squares regression, which is a joint classification and latent factor discovery approach. And then it's already very commonly used for other fields like chemometrics or brain imaging analysis, et cetera. And it's used to decode patterns, hidden patterns in the data. So partial least squares discriminant analysis is just a variation of the PLSR, which is more geared towards classification. And so that's what we are going to use. And the idea is like here, it tries to find the latent representation of genes that has a maximum covariance between the disease state and the gene expression signatures. And so we can isolate the gene expression patterns that would be most informative in predicting the disease context as well. So we first of all have to find these transcriptomic data sets for Alzheimer's and Parkinson's. So we look at the open source availability, and we have two data sets. So we have the RASMAP data sets for Alzheimer's, and for Parkinson's, we have COMMA data set. They have like 48 participants for the RASMAP from the prefrontal cortex, and for the Parkinson's, we have 12 participants, and they sample from the midbrain of patients because that's where most of the Parkinson's is thought to occur. And they are independently collected. They have been age and sex matched independently, so that and they also have strict quality control, filtering processes. All the single cell standard pipelines have been done by the authors. Even the cell type and annotation has been done by the authors. So it's a very clean data set, and we just take those data set right from the internet and we use it for our purpose. And then what we do is we take the Alzheimer's data sets in the beginning, and then we fit our PLSDA model to that. And then we extract multiple gene modules per cell type in the Alzheimer's data set. So we would have multiple gene modules for neurons, and then multiple for macula, multiple for astrocytes. And then we do some kind of post-processing. So for the post-processing, we are essentially looking at whether the modules we have are significant and the genes that are there are robust. And then we do the same thing for a Parkinson's data set. And this is separate again for each cell type individually. We extract multiple gene modules. And then we go in and we correlate them to see what is the level of overlap between the different gene modules that we have extracted. And so what we find is encoded in this heat map here. So we see that there is indeed a lot of overlap that is going on. So the size of the box here represents the number of robust genes that are shared between two gene modules. And the color, the intensity of the color represents what is the level of like similarity, or like how the genes are ordered in the gene module, how much those are similar to each other. And we see a lot of things going on. For example, the highest, like the most number of genes that are shared are between neurons in Alzheimer's and Parkinson's, neurons and oligoneurocytes in particular. And they have even like good amount of correlation with each other. And then we also see like glial cells, they have a lot of association with other cell types, like Alzheimer's glial cells have a lot with Parkinson's glial cells, and so on the Parkinson's as well. And then what we are doing is to, like, we have gene modules, sure, but how are they, like, whether they have meaningful information or not. What we do here is we look at the GWAS genes that have already been identified by other studies based on different techniques, right? And then we have GWAS genes on the x-axis. And here we have all the different gene modules, and we are seeing like whether these gene modules have any of these GWAS genes robustly associated in them. And these are the predictive weights from the PLSDA model. And so the very cool thing here is that, so the top are the Alzheimer's modules and the bottom are the Parkinson's modules. And here we can see, like, for example, this is ApoE gene from Alzheimer's. And the highest like the highest predictive weights are automatically in astrocytes and microglia. And this is just derived in a very data-driven manner. We haven't done anything to, like, make like astrocytes and microglia show ApoE presence. So that kind of gives us a confidence in our analysis. And similarly, we can see for the LRRK2 gene, which is one of the very important genes for Parkinson's, show up especially in microglia. And that has also been found in studies like very recently, 2022. And then it's not just that. The more, like the thing that we can go from here is that we see associations across, like, for example, these are Parkinson's genes, but Alzheimer gene modules also show high correlations or predictive weights for Parkinson's genes and vice versa. So some Alzheimer's genes, GWAS genes, show up in Parkinson's gene modules. And both, like, for example, we have just picked the first most important genes, and we have found studies like separately, which are very more targeted approaches. They have found similar kinds of support for finding ApoE in Parkinson's or SNCA in Alzheimer's, which are typically not found in GWAS analysis. And then to concretely understand what these gene modules mean biologically, we perform gene set enrichment analysis, which is essentially mapping different lists of weighted genes to predetermined gene sets, which encode information about biological processes and molecular functions, etc. And then we see like these broad kind of patterns emerge from our analysis. So we have like different glucose metabolic processes or cellular response to metal ion, etc. So for here, like each dot represents, so each node represents a term, a GO term, and the color represents whether this term was enriched for Alzheimer's or Parkinson's or both. And then like all these terms, like these were already, like there was literature supporting all of these broad terms, and people were like, oh, okay, there should be inflammation in both Alzheimer's and Parkinson's. And so these already naturally come up from our analysis. Okay, so I'm going to skip all this, and then we just did a comparison with the legacy approach, which is the differential gene expression, and we showed that that is not able to extract the amount of information that we got from our gene module level analysis. So that we have a technical replicate where we get similar analysis based on differential gene expression networks. And then to see whether this is replicable and whether it's just a bias in the transcriptomic data set, we repeated the entire analysis for 2 separate data sets, and we find very similar results. So yeah, in conclusion, we do show that there is a lot of overlap in Alzheimer's and Parkinson's, and we uncover the genetic mechanisms in terms of the gene modules. So it's essentially in terms of weighted gene lists. And then we see like the gene set enrichment analysis reveals the processes that underline these gene modules. For example, we have cellular response to stress, protein misfolding, glucose metabolism, etc. And then we verify our findings in a lot of different ways, like replicability analysis and validation tests, etc. So yeah, that's what I've been doing so far. Thanks.
Thanks, Anwesha, for the great talk. We are behind the schedule, so only one question.
Thanks, So how would you approach the fact that maybe what we're seeing is an attempted rescue response that looks similar in both pathways instead of a similar pathology?
Right. So yeah, that's a very cool question, and I have this plot here. So exactly to address that, what we were like, oh yeah, sure, there could be some pathways which are more like cell injury response, right? So that's why so in this analysis, what we did is we are looking at pathways that show up just specific to one cell type. So that kind of makes sense because like other cell types won't have that pathway. So maybe those are, because pathways which are related to cell injury would be probably shared across multiple cell types and gene modules. So we're looking at gene modules with pathways which come up in only one gene module. And then we look at what is the level of overlap across them. And then we see like neurons clustered together and then glial pathways clustered together. And those are more like immune inflammation. And here we have synapse pruning and different kinds of pathways come up here. So sure, we do have cell injury representation in the gene modules, but they also encode more intricate details that can be seen in here.
Would that also include compensation about neurons per se? If the neighboring neurons are dying, that one specific neuron kind of boosts all of this input?
Yeah, I mean, I'm not sure how we can go to that level of granularity, but I guess if we have like more data, we can extract, because here the data set is kind of limited in this size. So the number of components that can be extracted is also less. So the more number of components that we have, the higher granularity and higher level of details we can extract. And then, we can do all kinds of analysis to filter them out and look at exactly maybe what neurons are doing together or glials are doing together, stuff like that.
Thanks.